Okay. You can judge from my title that I here suggest where important experiments might be done by someone else. As a matter of fact, when I got retired some almost 20 years ago, I gave up also experimental work. So I wouldn't be able to do it myself, but it is an interesting question, as you will see, I hope so, from my presentation. In the first projection, this is a reminder to you that genetics by Mendel, Darwinism, of course by Darwin, and nucleic acid biochemistry by Miescher have their roots around 150 years ago. In the first time after that original discovery, all based on phenotypes. No one knew that there are genes. Then very little advance came. One just found out that probably the nuclei are important for inheritance. And it is microbial genetics, which officially started only in the 1940s, that could solve these questions which had not found with eukaryotic organisms, plants and animals, any good answer. So let me see. How do I proceed for the next? Oh, that's it. Yeah. Yeah, good. Thank you. It's like that. Yeah. You see here that it's around 1940 that people realized that bacteria and also bacteriophages, which are the viruses of bacteria, have genes and can mutate. And therefore, genetics became possible. And I list here 3 fundamental experimental approaches. One is all are based by taking 2 related bacterial strains, but having different phenotypes. And you take one as a donor and the other as a recipient, and hopefully you can show that some genes pass over from the donor to the recipient. Transformation is take from the donor, you destroy that cell, you extract from the extract, you mix this into recipient culture bacteria, and it is Avery and McLeod and McCarthy in 1944 who could show that only DNA from the donor does that, give transfer genes into a recipient. No other fragmentation which was made at that time. Almost no biologists believed that, they couldn't understand, but it was then later on a big help in 1953 when Watson and Crick published their double helical model of the DNA as a linear molecule. It is, however, already before that in the second bacterial genetic approach, taking the donor and mixing live donor with live recipients. Some bacteria do that, not all, that DNA is transferred, and interestingly, in this situation, very often, in a linear fashion, many genes which are there. You could break that couple by shear forces and show that after 1 minute there are only a few genes entered, later on after 10 minutes or more, and so on. So that indicated that these bacteria, most of these were then later on E. coli bacteria, have actually practically all their genes on one macromolecule of DNA. The third approach was that viruses which grow in a donor sometimes can incorporate DNA from the donor and transfer it by infection to the recipient cell. There are various ways to do that. Some bacteria viruses take just a fragment and occasionally take a fragment from the genome of the bacterial host and incorporate that fragment, just fill the particle with host DNA. Other viruses pick up a shorter segment and integrate that in the viral genome, leaving back often some viral genes, but one has studied those which are still able to replicate. And that gave the idea of gene vectors, that, for example, this virus has all the genes needed for replication, but not all the genes for producing particles afterwards. For example, the coat genes are missing, and that was the model later on, in fact, for genetic engineering. But I don't want to go into that matter. I go further to just remind you that various factors limit actually the gene acquisition in these three processes. One is the surfaces of the, particularly of the recipient cell, and in the conjugation of the donor cell must be able to do that. In virus infection, the virus must be able to adsorb to that surface and so on. Then if the DNA goes in, often bacteria are able to distinguish their own DNA from incoming foreign DNA. And this is called the restriction modification system in bacteria. And in these cases, the incoming DNA is rapidly degraded by endonucleases and then afterwards by exonucleases entirely. But during that very short period of time, sometimes a part of that incoming DNA finds its way into the genome of the recipient cell and acquires genetic information from the donor cell. Then, if that is there, it's a question if these genes are expressed, is the functional compatibility in order? That is kind of selection level for the propagation and for the expression of the genes, whether everything still functions as a replicating cell. And from these situations, you see that natural strategy of acquisition of foreign DNA in small steps is followed. You never get an entire genome which is not compatible with the genome of the recipient cell. As a follow-up of all these investigations, me and other colleagues started to wonder, in fact, yes, of course, as I mentioned, Darwin looked at phenotypes, and Darwin spoke on variants. Now then, we could start to talk on genetic variants, and question was, what are the molecular mechanisms to make a mutagenesis in the bacterial genome? This is a summary of our knowledge. On top, I remind you the classical pillars of evolution, biological evolution. Evolution is driven by having occasionally in one cell of a large population a mutation, making a genetic variant. Then all novel mutants are submitted to Darwinian selection, whether they get well along with the encountered living conditions, And finally, there is also, of course, isolation, which Darwin had described, and I don't go into detail for that. The question is, okay, on the lower left, I summarize that there are a multitude of different mechanisms, many already identified mechanisms, which give occasionally a genetic variant. Some are due to occurring during replication. These are often local mutations, as such as nucleotide substitutions. Then there are rearrangement processes that a part of the genome becomes duplicated, as a part of the genome, a segment becomes deleted, a part gets transferred. We talked just before on transposable elements, and then also some elements can invert on the site where they are. So these are internal rearrangements, and in the meantime, there are also mutagens, as we know. These can be chemical substances or radiations, and they can give, depending on what kind of action they cause to the genome, local mutations or DNA rearrangements within the genome. The third big strategy, natural strategy, is horizontal transfer. In one of the talks, this is equivalent to lateral transfer of a segment of DNA from a donor into a recipient. And this is now what I then later on, you know, when the microbiologists started to talk on that, and lots of research was done because horizontal transfer is very important for medical problem of horizontal transfer of antibiotic resistances. But let me go to a consensus of this field of mutagenesis. In fact, favorable mutants which give selective advantage are rather rare. More often you see immediate influence on life processes, even sometimes lethality, and these are contraselective. Then a number of mutations have no effect because they do not kill a gene or are not at the place where they would kill expression control element or so. So from that kind of range of possibilities, I conclude that there is no good evidence seen that the bacterial cell knows which gene to change in which order. It's more a random chance that mutagenesis brings an advance or not an advance. However, these frequencies must be low. Nature masters to keep the mutagenesis rates very, very low. It's very protective to the most of the genomes in the growing population. And this is remarkable. I'm sorry, I forgot to. That's what I just explained. I think it was clear enough. The next thing is that the contributions to biological evolution of these three strategies, local, internal rearrangements within the genome or acquisition of a segment of DNA, have different, qualitative contributions to evolution. Let me go just to DNA acquisition, which is on the right-hand side. That's a sharing in successful development made by others, and that is what for a long time scientists working with eukaryotes said that's only occurring with bacteria. They studied that, and it's interesting, but it doesn't occur in higher organisms. However, one must be aware that all these processes are very, very rare, and therefore it's not so easy to see it. When then around 2000, the human genome was published for the first time, one of the interpretations was in that human genome, they identified in the order of 150 or so bacterial genes. Of course, I'm not sure, I cannot remember which bacteria were compared, but in some way we need more data for that. Why? Because you see that Darwin already have drawn the blue tree of evolution and concluding that the living beings which are on the top, today's living beings, have common origin. He doesn't say whether one or many different origins, and I do not know that because, of course, we have no idea so far how the first living origin started on this planet. However, we do know that this tree is correct as far as we can say, and occasionally these connectors, which I just randomly have thrown into that tree, shows that occasionally a gene can horizontally transfer and, under particular conditions, give an advantage to the recipient. And my conclusion is Darwin was right. Common origin is fine, but with the horizontal transfer, all living beings have also a common future. And therefore, that's for me quite important that we take very big care for the big biodiversity, because if we would destroy the biodiversity, then all of these may be still very important genes for future developments in an ever very slowly developing non-living world. Different habitats show up. We must be able to adapt, and we should avoid to destroy that nature. And for this reason, it is important that more research would be done as the one of just comparing the genes in the human genome and in, I'm not sure, probably not too many bacterial genomes, because, of course, with the microbiome, on which we do know that there are more bacterial cells both in my bodies and in plants than higher plants of diploid shape. And living cohabitation favors occasional gene transfer. But very rarely. And nature knows also to keep these gene transfers low, and I mentioned already some of these barriers before. I finish with my hope that in future ways, good on the more and more becoming available data on many microorganisms and on many plants and animals and humans, we could see, particularly for traces of homology, and homology is not necessarily absolutely identical sequences, but there may be near identical sequences. And it would be interesting to compare then, are these sequences found both in the human genome or in the plant genome and in bacterial genome? Are they bacterial origin, or did the bacteria pick up when they grew in their host plants or human or animals some of these genes? So it can go both directions. So many things are open for future investigation. Before I finish, time has over, but let me see, I would like to just make two interesting conclusions. For me, these genes which help evolution, I call them evolution genes. They can be variation generators by recombination enzymes favoring recombination occasionally, or by other ways to create the mutation. Then there are gene products which act as modulators of the frequency to keep the frequency at very tolerable low level. In bacterial systems, per generation, you have perhaps a double mutation per more than 1,000 cells in the population, 2,000 or so. Then you conclude from that gene elements are there, and also nature uses non-genetic elements, which can be, I give you just one example, which you can, which go back to Watson-Crick. They notice that very short-living isomeric forms of nucleotides, for example, adenine, in an isomeric form in which one nitrogen atom is not at the same place than in the most stable form, cannot pair with its normal thymine, but by chance it can pair with cytosine. And in all textbooks which I looked at, this is called an error. For me, this is not an error. Nature uses these things in order to very rarely, occasionally produce a mutation. And actually, there are repair enzymes which try to keep that rate even very low and tolerable. Then the conclusion is natural reality takes actively care of biological evolution. And the next final conclusion from all that is that we have experimental data. Some of these evolution genes in E. coli, we have mutated them that they are not active any longer, and these cells grow absolutely perfectly with the same generation time, no problem. So that shows that these evolution genes are not at all needed for the life, during the lifetime of this individual. And my extrapolation is that in my body, in my genome, I have also evolution genes, but they are not responsible for my own fulfillment of life, but they contribute to the expansion of life by steady, very slow evolution, and therefore this is the source for biodiversity. Thank you for your attention.